Welcome to Introduction to Biology. In this lesson, we will discuss Mendelian genetics. In it, you will learn Gregor Mendel's experiments which laid the foundation for modern-day genetics. Genetics is the study of heredity. Heredity, it means the transmission of traits or the passing down of traits from one generation to the next. The three main learning objectives of this lesson are to understand Mendel's experiments and the laws of probability that govern this, the characteristics and traits from one generation to another and how they're passed down through the laws of inheritance that Mendel was able to figure out. So what are we going to learn about today? We're going to try to understand what heredity is. We're going to describe the scientific reasons for the success of Mendel's experimental work. We're going to describe the expected outcomes of the monohybrid cross involving the dominant and the recessive alleles. We're going to explain the relationship between a genotype and a phenotype, and we're going to understand what incomplete dominance is. And then in the final thing, we're going to understand how to describe and understand the pedigree chart and what it says to us. There are many misconceptions historically about what heredity is. They thought, for example, the sperm contained a fully formed little person, or that the offspring were like their parents, and like begets like. Everything came from the egg, and the females controlled all the traits. Other misconceptions were that males controlled all the traits, and that offspring were the average between both parents. Inheritance, acquired characteristics, changes happening to parents could be passed on to offspring. And the idea that particles called gemmules carry the traits we inherited. So there were many different types of misconceptions before Mendelian genetics came along and started to foundationally put some understanding behind the science of inheritance. For example, in the old days, this is found from 1640, a Swedish text, that there was a fully formed human being that was found in a sperm. And some earlier editions even had clothed little men and women in fashions of their day in the sperm. So, and that's how things were passed on. In terms of genetic terminology, we need to understand that a trait is a characteristic that can be passed from one parent to another, and genetics is the study of heredity, and heredity is this passing out of traits from parent to offspring. So it's the passing down from the dad and the mom to the various boys and girls that they have, their children, various characteristics that get passed on from generation to generation. And each version, there's two versions of a gene because mom has a pair of chromosomes for each trait she got from her dad and her mom. And each copy doesn't have to be the same too. We have different versions of the same copy as can be seen here. So the father can be having two versions and the mother can be having two versions of the same gene, and they can be passing it on to their son or daughter in terms of whether or not it is a dominant gene or recessive gene for a particular type of characteristics. And here we're talking about the whole idea of getting citric fibrosis. So who was Gregor Mendel? Gregor Mendel was an Austrian monk. He studied 28,000 pea plants from 1856 to 1863. When he was working in the 1860s, he found that parents passed on discrete genes, what he called heritable factors, onto their offspring. Genes are responsible for inherited traits, and these genes retain their individual identity generation after generation. These were the main three things that he found that we'll go into. He is considered the father of genetics because he led the foundation of modern-day genetics. He understood that genetic crosses that are governed by the rules of probability and the rule of multiplication, which states the probability of a common compound event is the product of separate probabilities of independent events. He had a strong mathematical background to be able to understand these patterns of inheritance and make sense of his 28,000 crosses. His family farm in Austria gardened and did some beekeeping. He was sick. He was kind of sickly in the farm, so his brother took over. He went on to the university to study philosophy and became a friar to pay for his education. He then entered the Augustine St. Thomas Abbey and trained to become a priest and taught science. He was sent to the University of Virginia to study under the sponsorship of Abbot C.F. Knapp. When he returned to the abbey, he taught a while studying heredity and eventually became an abbot and gave up science. So why did Mendel pick peas? Well, there was numerous reasons that the pea plants were a very good pick for him. They were simple dichotomous traits. That is, they had traits like being a flower color or a shape of a seed or a seed color. And these traits could be used in true breeding experiments. That is, plants that didn't have any traits from other plants. And so basically plants that were purebred. So, and they had short generation times. So peas were a new individual. They could value for thousands of offspring. They were cheap, readily available, and they could be used in short generations for Mendel time. So Mendel's notation system is capital P is for the parental generation of true breeding plants. F1 is the first filial generation. F2 means the second filial generation, and so forth. F3, third filial generation, F4, etc. succeeding generations. The siblings are brothers and sisters. Cross siblings are matings between the brothers and sisters. The back crosses are matings of offspring back to their parents of the other ancestor. A self-cross is the crossing of a plant with itself, and a true breeding plant is when plants have the same traits always produced and have the same traits.